Okay, so this is my updated Unit 4 review PowerPoint or video for the 2025-2026 school year because College Board made some changes to Unit 4. And so let's go ahead and do this. So when we look at the text in this video, in the colorful font at the top of each slide is the objective that College Board is expecting you to know, to be able to do, to write, to describe, explain, etc. And then in the black text, that is going to be the essential knowledges, copy and pasted straight from College Board. So this is an overview of Unit 4, and I am going to go kind of fast because it's meant to be review. If you need to understand any of these topics in more detail, I recommend hitting pause and then checking out my Unit 4 playlist where I have each of these topics in more detail. Okay, so let's begin with cell communication. So cells communicate in a variety of ways. We can have direct contact, or direct cell-to-cell contact. This is very popular in our immune system with how our immune system cells communicate. We also see this in plant cells where they have these like channels or like openings in their cell, not really openings, I'd say channels or gaps in their cell walls where molecules can travel directly from one cytoplasm to the next in a nearby cell. And then we have short distance communication and long distance communication. And so in short distance communication, this is where cells are going to send out chemical signals, but they don't travel in the bloodstream. They travel just a very short distance to adjacent or nearby cells. We call these types of chemical signals local regulators. And so you can think about if you have like a cut on your skin, the damaged cells are going to send out chemical signals called growth factors that will signal to nearby cells who receive that message, hey, we need to enter into the cell cycle and divide and repair that wound. We also see this in our nervous system where we have a neuron, a presynaptic neuron, and then it releases neurotransmitters into that synapse. That's a very short distance for those neurotransmitters to travel. So those would be examples of local regulators. And then we have hormones, and this is really involved with our endocrine system where we have glands. Glands are organs that release hormones, and they're going to travel throughout your entire bloodstream. So our circulatory system is like a transportation system, so hormones released into our bloodstream will travel everywhere. They won't have an impact everywhere. It just matters really for target cells. Target cells are cells that have receptors for a particular chemical messenger. Okay. Now, when we talk about, well, what happens when a cell receives that signal? So we have 3 steps to basically cell communication. There's reception, when it receives the chemical messenger. There's a transduction, where that message is basically converted to something intracellular. And then there's a cell response. Cell response could be something changing the cell's behavior or changing gene expression or apoptosis, which is programmed cell death. But they generally have 3 steps. So if we look at our essential knowledge here, signal transduction pathways, which are the intracellular part of cell communication, and really use a lot of proteins, different kinds of proteins in there. Signal transduction pathways link signal reception, that means like receiving the signal, with the final outcome or the cell response. What was that signal intending? For example, here we have insulin. Insulin is that chemical messenger, that hormone, and it is received by a protein receptor. That's going to then activate a signal transduction pathway and a series of changes within the proteins within the cell that bring about a cell response. In this case, it would be to move protein channels to the cell membrane so that way glucose can enter into the cell or into the tissues and be removed from the bloodstream. Okay, now let's go ahead and talk about that transduction step. So chemical signals like hormones or local regulators are generally called ligands. So when a ligand attaches to a protein receptor, here I have probably epidermal growth factor, EGF. So when that ligand attaches to that protein receptor, that's in blue here, that's going to cause a conformational change in shape. On the intracellular portion of that protein receptor, it's going to change shape, and that can activate a relay protein to then further cause changes within the cell. A lot of the changes within the cell in the transduction step rely on protein modifications and phosphorylation cascades. So a key type of protein in this is called a kinase. And kinases are responsible for phosphorylating other molecules. And when we phosphorylate a molecule, that causes it, usually a protein, causes it to change shape, go through a conformational change in shape, and most of the time is going to activate that protein. There are rare cases where a phosphorylation of a molecule will deactivate it, but generally activates it. Now, if you're thinking, well, why? Like, what's happening? If we remember, a phosphate group has a negative charge. So if you add a negatively charged molecule to a protein that's made of R groups, those R groups of those amino acids, and the protein is made of amino acids that have R groups. Those R groups have positive, negative charges, polar, nonpolar chemical properties. So when that negatively charged phosphate group binds to that protein, it causes shifts in how those R groups have attracted or repelled to each other, and that leads to a conformational change in shape. Okay, now signaling begins with the recognition of a ligand, as I mentioned earlier. And so the ligand binding domain means that the extracellular part of the receptor protein is going to receive and recognize that ligand. It can be a protein or a small molecule. An example of a type of protein like this are G protein coupled receptors, as you see in purple on my screen here. Now, when we talk about signaling and different types of ligands, we have ligands that are sterols or steroid-based hormones. Now, those, if you remember from Unit 1, are nonpolar hydrophobic molecules, and they can actually diffuse directly through the lipid bilayer and the nuclear envelope. And so their protein receptor is either going to be in the cytoplasm or in the nucleus. Now, regardless of where it's located, most steroid hormones are going to have a direct cell response when they attach to the receptor protein of activating gene expression. Now, protein-based hormones, on the other hand, cannot diffuse through the lipid bilayer. So their receptor protein has the ligand binding domain on the surface of the cell. These are often called cell surface protein receptors, and those would be for your protein-based ligands. These are going to rely on a transduction pathway within the cell and protein modifications to pass on or relay that signal that was sent from the original ligand. So when ligands attach to receptors, especially if you're using a second messenger, I don't really show it a lot in my diagrams, but on the inside, that signal is being amplified. And so if we talk about like a G protein linked receptor, a cyclic AMP is produced, which is a second messenger, which we'll see coming up here in a couple slides. It's not just one second messenger produced like I show in my homemade diagrams, but it's actually multiple second messengers produced that activate multiple phosphorylation cascades, and the signal amplifies within the cell. Okay, so after the ligand binds, we can use epinephrine in the top. When the ligand binds, the protein receptor undergoes a conformational change in shape. So in that top circle, you can see on the left with no ligand versus on the right with the ligand, I have a slightly different shaped receptor. So there's a conformational change in shape that then activates proteins within the cell. Down in the bottom example, we see a receptor tyrosine kinase. When the ligand attaches to that ligand binding domain, those two receptor polypeptides form a dimer, and now it's an active receptor tyrosine kinase that will then phosphorylate itself. Okay, sorry, it's not active right when it forms a dimer, but when it forms a dimer, now it can phosphorylate itself, and then it will be an active intracellular receptor protein. Okay, now when we talk about what's happening on the inside, there are some types of signaling that rely on second messengers. A common second messenger is cyclic AMP. So when the ligand attaches extracellularly, and in this case we have a G protein that becomes active, it will activate adenylyl cyclase, which is the pink square on my screen. That's an enzyme that will then convert ATP into cyclic AMP, which is our second messenger. Cyclic AMP is now going to go activate the phosphorylation cascade. It will activate pKa. Now, cyclic AMP does not stay active forever. It will actually, it has a different enzyme that will come and deactivate it, shutting off that phosphorylation cascade. Okay, now if we talk about hormones, hormones are a type of ligand or signaling messenger, and they travel long distance in our bloodstream. So they only have impacts on target cells that have receptors for them, but they are sent out into our entire bloodstream. Now, another type that we see in the nervous system are going to be ligand-gated ion channels. And so ligand-gated ion channels are a type of receptor that when the ligand attaches, it opens up that gate and ions can flow in. This is related to the depolarization of the membrane that I kind of talked about in my Unit 2 review and topic for 2.6. So it really has more to do with the nervous system, but these are ligand-gated ion channels that can change the overall charge within a cell depending on if the ions are flowing in or out, or if they're positive or negatively charged ions. Okay, signal transduction pathways may result in changes in gene expression, which we can see in the top right up here. The genes, sometimes the final cell response is to phosphorylate and activate a transcription factor, and that would turn on gene expression. It could be changes in cell function, like we saw with insulin. That's going to put protein channels into the cell membrane to have facilitated diffusion occur and bring glucose into the cell. It may also result in programmed cell death or apoptosis. If we have an unhealthy cell or a cell with damaged mitochondria, sometimes there's a signal where the cell is not repairable and we need it to go through a programmed cell death. That will be apoptosis. Now, changes in signal transduction pathways can alter cell responses. So I mentioned earlier that each of these molecules are proteins. Proteins are coded for by genes. Genes are DNA where mutations happen. So if there's a mutation, let's pretend there's a mutation here in this SOS protein, well then the rest of the transduction will not be activated and there won't be a cell response. So mutations can happen in any step of cell signaling, and so it really depends on the situation. So if you have a test question about that, pay attention to context clues. Chemicals that interact with any component of the signaling pathway may activate or inhibit that pathway. One that I find particularly fascinating is metformin. So people with type 2 diabetes may take metformin as a medication. Now what metformin does is it actually activates a different little tiny pathway called the AMPK pathway, AMP kinase, which will actually increase the sensitivity of the insulin receptor. So by making the insulin receptor more receptive to insulin, now the cell can respond and help bring in glucose. So metformin would be a chemical, like a medication, that is used to help people with type 2 diabetes. Okay. So now we're moving into feedback loops. There's only two essential knowledges when it comes to feedback. So negative feedback mechanisms are used to maintain homeostasis by reducing the initial stimulus. A common example of this is going to be insulin and glucagon with blood sugar balance. If your blood sugar levels rise, that stimulus is so large, we're going to use insulin to communicate with our cells to bring our blood sugar levels back down. And if our blood sugar levels drop too low, we're going to use glucagon, the other hormone, to bring us back up and reduce that stimulus. So negative feedback loops are used to maintain balance. And I have a whole video on negative feedback loops with multiple examples that I recommend. Positive feedback loops, on the other hand, do not return you to homeostasis. Instead, they amplify. And the perfect example of this is childbirth. Contractions get stronger and stronger and stronger and stronger until the baby is born. I have a whole video on positive feedback loops with multiple examples that I recommend you watch. Okay, now our last thing to talk about is the cell cycle and cell cycle regulation. So the cell cycle is a highly regulated series of events that control growth and reproduction of eukaryotic cells. So we're going to talk first about interphase. Interphase is made of G1, S, G2, and then we'll talk about M phase, which is composed of mitosis and cytokinesis. So they all happen in a very controlled series of steps. So in G1, the cell's metabolically active. It's carrying out its life's functions, making organelles, cytoplasmic components. For example, my skin cells, if I go out into the sun with UV light, the cell response would be to make some melanin, right? I'll get tan, I'll get a little bit darker. That's my skin cells carrying out their functions. Those are cells in G1. Now let's say my skin cells get cut and they receive a ligand, like a signal, a growth factor, a local regulator. Hey, we need to divide. Well, now the cell receives a signal and it's going to go into S phase, which is where only cells that are going to divide enter. So a cell that isn't dividing will actually exit the cell cycle and go into G0, which you see here, and we'll talk about in a little bit. But if a cell is going to divide, it will pass that G1S checkpoint and move into S phase, where our DNA is replicated. On my screen here, you see an unduplicated chromosome and then a duplicated chromosome. They are both considered to be one chromosome, but in S phase, that is where our DNA is doubled. And then we go into G2, and G2 protein synthesis occurs. A lot of ATP is made in preparation to go into M phase. M phase is where the cell actually divides, and you can imagine we're dividing an entire nucleus and then separating the cytoplasm. That's going to take a lot of ATP. Now, centrosomes are going to replicate during G2. Centrosomes are what eventually send out spindle fibers during M phase. Now, a cell that's not going to divide or is in a resting state will enter G0, G0. So non-dividing cells, like your brain cells, will never go through mitosis. They may spend 65, 70, 80 years in G0, just carrying on with their daily functions. Sometimes cells like your liver cells generally are in that resting state and will only get called back to the cell cycle if there's damage. Okay, now I do want to point out, if a cell receives a signal to divide, that G1S checkpoint is super important, and if there is too much damage and the cell can't divide, it'll be held and then go through apoptosis. Okay, I think that's why I have that there. Now, mitosis is a process that ensures, like when we go into M phase, that a complete genome is passed from one cell to two daughter cells. So the chromosomes are replicated chromosomes. We've gone through DNA replication, so we have twice as much DNA as we need. Those replicated chromosomes are distributed and separated into daughter cells, and those two daughter cells should end up identical if everything happens correctly. So mitosis is important for growth and repair, but also asexual reproduction. So mitosis happens with prophase, metaphase, anaphase, telophase, and alternates with the cell cycle with interphase. So prophase, if we look at it first, so here on my left I have a cell in G1 in interphase. In prophase, the chromosomes have already duplicated in S phase. Now I have identical sister chromatids held together by a centromere. What I do see is the centrosomes have started to send out spindle fibers or microtubules that are in green. Those microtubules during metaphase will attach to the centromere. There's proteins on the centromere called the kinetochore where the microtubule spindle fibers will attach. This will ultimately have our duplicated or replicated chromosomes line up along the middle or the equator of the cell. Then in anaphase, the spindle fibers will begin to pull apart those sister chromatids. So now we have unduplicated chromatids or individual chromatids. Technically, they should be called individual chromosomes at this point. And then in telophase, new nuclear envelopes begin to form. Our DNA uncondenses and goes back to chromatin. And then eventually cytokinesis will happen where we'll separate that cytoplasm. In animal cells, there's a cleavage furrow that forms, while in plant cells, it's a cell plate. A new cell plate is formed between the two nuclei. Now, we do have checkpoints to control that all of this happens smoothly. At the G1 checkpoint, we check for DNA damage. Is there enough nutrients? Is the cell healthy? Is it healthy DNA? And if it's a go, well, then the cell will enter into S phase. If it's not, then we can put it at on hold, I guess, and then we could try to fix the DNA damage. And if it's irreplaceable, not fixable, I don't know, then the cell can undergo apoptosis. Now at the G2 checkpoint, we're going to make sure that all replication is finished and all of the chromosomes are in their duplicated forms. And then at the M phase checkpoint, that's going to make sure that spindle fibers have attached to every centromere so evenly so that sister chromatids can get pulled apart. If there's no spindle fiber on one side, then the sister chromatids would stay together, and that won't be a good thing. During the daughter cells will have not equal numbers of chromosomes. Now, how this is regulated is through cyclins and cyclin-dependent kinases. As I mentioned earlier in the video, kinases phosphorylate and activate other proteins. As we can see in this picture, the kinases, the cyclin-dependent kinases stay constant throughout the life of the cell cycle, and it's the cyclins that vary in amount. So at different stages of the cell cycle, we have different proteins becoming phosphorylated and different proteins becoming activated. So in S phase, for example, that S phase cyclin attached to the CDK will activate replication enzymes by phosphorylating those enzymes, and now polymerase 3 and helicase, DNA polymerase 1, primase, etc. can replicate our DNA to make our identical sister chromatids. But we don't want replication happening forever, so eventually that S cyclin degrades and replication is finished. So it's cyclins and cyclin-dependent kinases that regulate which phase of the cell cycle the cells are in. Now, disruptions to the cell cycle may result in cancer, which is abnormal cell growth, and it often is characterized by a faster or shorter cell cycle. They are just speeding through the cell cycle. And so if we talk about this, we have a couple important types of genes. We have tumor suppressor genes that you can see on the left of my screen are meant to inhibit. So a tumor suppressor gene is a gene like P53 that is going to say, hey, stop, the cell is damaged. We need to fix our DNA before we go into S phase. And this is a good thing. We want to stop abnormal cells from dividing. Now, if we can fix the DNA damage, well, then the cell can go into S phase and divide. If we can't repair the damage, then hopefully the cell goes through apoptosis. Now, if you look at the diagram like in the middle, if there's a mutated tumor suppressor gene, like P53 is mutated and cannot work, well, then there's nothing, or generally nothing, to stop the cell cycle, and now we have uncontrolled cell growth. And so that could be like when people talk about like the breast cancer being inherited, that's because the BRCA gene, the BRCA gene, is a tumor suppressor gene. That when it's working normally, it stops abnormal growth in breast tissue. However, if it's mutated, now there can be uncontrolled cell growth because it's not functioning to stop the cell cycle. Now, the other vocabulary that's important is the word proto-oncogene. A proto-oncogene is any gene that codes for a protein that controls the cell cycle. So that could be a growth factor receptor, could be a part of a protein in the transduction pathway, it could be a cyclin. So any protein involved in cell cycle regulation and cell division. The normal functioning version of that gene is the proto-oncogene. Now, if there's a mutation in it, and now maybe it's a gain of function, it's hyperactive, it's sending too many signals, and now that's leading to uncontrolled cell growth, that would be now called an oncogene. And that's why like the cancer department at a hospital is oncology. So an example of this would be the RAS protein in the growth factor pathway, where the RAS protein it has a mutation where it folds up to be overactive. And so now it's just sending a signal to divide, even though there's no ligand. So that would be a gain of function mutation that causes the cell cycle to keep on going. Okay, so that is my review of Unit 4. And great job and good luck on any test you are studying for.